All right, thank you for the opportunity. So basically the question we're asking here is, are individual resting fMRI connectivity patterns unique across different runs? And can it be used to identify individuals, pair individuals between runs? So I'm going to try and use the mouse here. So a bit of a background. So individual differences in resting fMRI and the functional connectivity to kind of act as a fingerprint was introduced by Emily Finn in 2015 in a Nature Neuroscience paper. And in that work, they identified individuals based on resting fMRI functional connectivity across runs. But that study was limited to a small number of subjects. It was 126 N of 126. But in this study, we are trying to replicate that with a larger number of subjects, N of 820. And we're trying to answer the following questions. What kind of performance are produced by different brain atlases? Is the result robust across multiple runs of fMRI? The effect of temporal delay and the order between different fMRI runs, and FC difference between the correct match and the rest of the subjects. Is it due to FC or is it due to something else? And then are there genetic associations of FC? So the data for this project was acquired from the Human Connectome Project, the S900 release. And as you know, they are all from healthy adults. The age was between 22 and 37 years. And we picked 820 subjects. And these subjects were based on the fact that they all had all 4 runs. The release had more than 900 subjects. It contained about 366 males and 454 females. Now, as I said, there were 4 runs. Run 1 and Run 2 were acquired on day 1, and Run 3 and Run 4 were acquired on a different day, but on the same day. Now, they were all axial acquisitions, and the phase encoding was from left to right for Run 1 and Run 2, for Run 1 and Run 4, but from right to left for Run 2 and Run 3. So we used 10 different brain atlases here. The first 5 are functional atlases, and the next 5 are structural atlases. The DOS 160, the CADOC 400, CADOC 200, and so on. And here are some representations of these atlases. And they have different number of parcellations and nodes in these atlases. So the method is quite straightforward, and it closely follows to the Finn et al. Paper. So for each region of interest, we find the average time course, and then we compute the Pearson correlation between the regions of interest to come up with the functional connectivity matrix. And then, so the FC matrix is a symmetric matrix. So after the removal of the upper half, we basically find the correlation between FC of different runs. Again, a simple Pearson correlation. So I'm going to call that correlation the FCC, the functional connectivity correlation. So here, for example, you have subject 1 from run 1. We compute its FCC with all other subjects' functional matrices from a different run. And the match is made by assigning the first subject to the maximum correlation, the maximum FCC. So here are the results. So these are the 10 different atlases, and the percentage, the accuracy of the correct identification as a percentage. And so we have 4 different runs, and if you combine them 2 at a time, you get 6 possible combinations. But you can also flip the order of the runs from pre to post or post to pre, so you get 12 to a total of 12 different combinations. And this is the average across all those 12 runs and the standard deviation. So as you can see, the DOS 160 performs at an accuracy above 90%, closely followed by the CC 400, and then AICHA, and so on. And the poorest performer was the Brodmann Atlas. Now, the previous talk mentioned about the functional structural relevance, and here you see that the functional atlases are performing better than the structural atlases. So in order to further investigate the a question of the temporal delay between the runs. We looked at these individual pairs separately for all the 10 atlases. And the best performer was from R4 to R3. So predicting R3 from R4, and this performed consistently at a high rate than the other runs. And the second best was R1 to R2, and the poorest was R1 to R4, or R4 to R1. So what this means is that it seems that the identification accuracy is higher when the temporal delay between the runs is shorter. So because R3 and R4 were acquired on the same day, as well as R1 and R2. And R1 to R4, or R4 to R1, has the highest temporal delay. Now, we also see differences between the pre versus post, or post versus pre. So there is a question, this raises a question, is it because of the temporal order, or is it due to the direction of the face encoding? As I mentioned to you before, R4 and R1 have left to right face encoding, and so I haven't answered that question yet, but needs further investigation. Now, we also checked whether the FCC difference between the correct match and the rest of the subjects is significant, right? So here, the FCC of the correct match is indicated by the blue dot, and the subjects are along the rows, and the FCC for all other subjects, right, is shown as a histogram for each individual subject, right? And the mean of that histogram is also shown as a blue dot. So we looked at this for the highest performing runs as well as the lowest performing runs. And across all these 6 pairs of runs, we see that the FCC of the correct identification is several standard deviations away from the mean of the others, which means that we didn't nearly closely miss the next guy, but the identification was quite accurate, like very far away from the rest of the subjects, for most of the subjects. And these differences are statistically significant. And this is true for the highest performers as well as the lowest performers. So the other question is now, is it because of FC pattern of individuals, or is it because of something else? And the first question we asked was, is it due to motion? So the data we used were after the ICA fix denoised. So the motion artifacts and the other artifacts were removed during that process. And then all the time series from resting fMRI were detrended, as well as 24 motion parameters regressed out. We also checked that the time series were uncorrelated with to frame displacement. So it seems that it's not due to motion, right? The next question is it due to global signal? So we computed global signal in 2 different ways. In the first way, we basically average the time courses for each individual subject. And in the second, we first normalize the time series and then average. And in both cases, after we regress out the global signal, for DOS 160, the accuracy of identification increases to 97 to 99%. So previously it was 91%, but now it goes up. So this indeed kind of tells that it's not due to a global signal. In fact, regressing out the global signal helps in the accuracy of identification. We also did a permutation test of FC where we fixed the order of FC of run 1. And then for the other run, we basically randomly shuffled the FCs, and the identification accuracy dropped to less than 2%. So here is a preliminary result on the question of, are there genetic associations of FC? So what we did here is we looked at the FCC difference between self and his or her twin pair, or self and his or her next best match for the non-twin guys. So there were 48 pairs of dizygotic twins, 93 pairs of monozygotic twins. And when we plot this difference between self and twin for these all 12 different possible combinations of runs, we see that the monozygotic twins, this difference is smaller than the difference of the dizygotic twins. And this result was significant for all 12 runs as well as the average across runs. However, when we looked at the next best match who did not have a twin, that difference was smaller than the monozygotic twin. So this could mean two things. One is that there may be someone out there who has a functional connectivity pattern that is closer to your twin, between you and the twin is larger than the other guy. Or this may be due to the fact that you have a very large number of non-twins here. So this needs further investigation as well. So to conclude, I think the data that I've shown today indicates that individual identification from resting fMRI functional connectivity is possible. We investigated the performance of multiple brain atlases, and DOS 160 was the best performer. Performance of different pairs of runs indicate the influence of time delay between runs. FC correlation between difference between correct and the rest was very significant, indicating that the identification was very accurate and far away from the rest. Regression of the global signal seems to increase accuracy, and some preliminary results indicating the presence of genetic association. So what's the take-home message here? I think as a community, we do a lot of group analysis, and I think we need to develop analysis methods that preserve individual differences. Now, a larger cohort and many more tests are needed to make functional brain connectivity closer to fingerprint, even though our rates are very high. And I have quite a few ideas, but would like to get your thoughts and hope I could talk to you after the talk or at the poster, which is 4081 on Thursday afternoon session. Okay, so acknowledgements. I would like to thank my lab, especially Chao Sang, who did some of the analysis. A special thanks to the Human Connectome Project for collecting the data and all the participants who participated. So with that, I would like to conclude. Thank you.
Great, thank you. We have time for one question. Does anyone have a question? Yeah.
Maybe I missed something, but why did you consider the Shet parcellation, since that's what was used in the thin paper?
So I couldn't find that readily available. I think they used a Yale, they referred to that as a Yale study. These were the ones that were publicly kind of available. I thought I should email, but I didn't email.
Just a quick remark that the Human Connectome dataset has quite a bit of siblings, like the 900 released is broken into 300 families.
Yep.
So it could be that the
so I'm aware of that, and also some of the twin pairs we used were the genetically tested. There are also other twin pairs that weren't genetically tested, and they were all included in the non-twin analysis. So that could be another reason why it's the difference is smaller.